Hello everyone. Our chapter today we are going to discuss is the introduction and the bacterial taxonomy. So, first of all, you should know the term microbiology. It was introduced by the French chemist, that is the Louis Pasteur. So, this picture suggests the French chemist, that is Louis Pasteur. So, who is known as father of microbiology? It's the Louis Pasteur. So, for examination point of view, you should know this point that Louis Pasteur is known as the father of the microbiology. Due to his groundbreaking work in the field of microbiology, he made numerous discoveries related to microorganisms. So Pasteur was the person who developed the germ theory of diseases. So what is germ theory? According to that, the many diseases, they are caused by the microorganisms. The Pasteur, what he did, he conducted experiments that provide strong evidence for the role of microorganisms in the fermentation and the diseases. So he laid the foundation that microorganisms, they could be responsible for the transmission of diseases. However, these microorganisms cannot be seen by the naked eyes. They are seen only under the microscope, and he termed them as animalcules. So that means they are very small living creatures that are not visible to the naked eye. So the germ theory of disease, it is scientific theory that states that the microorganisms, including bacteria, fungi and viruses, they can cause infectious diseases. However, this germ theory was not attributed to single individual, but it was developed over time through work of several scientists. Apart from that, he developed anthrax and the rabies vaccine. So, rabies vaccine, he developed the first rabies vaccine and the anthrax vaccine. Louis Pasteur also developed the pasteurization process that involves the heating liquids such as milk and wine to kill the harmful microorganisms without significantly affecting the taste and quality of the product. So, his work on the anthrax and rabies vaccine paved the way for modern vaccine development. Apart from that, he also made a significant contribution to the process of fermentation. For entrance exam point of view, you have to just remember that the Joseph Lister is known as father of antiseptic surgery. So this was Joseph Lister. So he introduced the concept of sterile surgical field. So who introduced the concept of sterile surgical field? It was Joseph Lister. Who is the father of antiseptic surgery? Remember, it's the Joseph Lister. Edward Jenner. Well, the Edward Jenner, it is best known for his work in the developing the smallpox vaccine. So he was Edward Jenner. So who is known as father of immunization? It's the Edward Jenner. So what he did was he used material from the cowpox. To inoculate the to inoculate people demonstrated that it could protect them against the smallpox. So he laid down the foundation for principle of vaccination. So that is why he is known as father of immunization. So what he did was cowpox inoculation. So Jenner conducted an experiment on remember May 14, 1796. He took material from the cowpox lesion on the milkmaid hand and he inoculated on 8-year child named James Phipps. So the boy developed a mild case of cowpox, but he recovered quickly. After observing James Phipps had developed immunity to the cowpox, Jenner then inoculated the boy with the smallpox material. So the James did not contract smallpox. So this experiment demonstrated that cowpox that could protect against the smallpox and laid to the foundation for development of the vaccine. So he played an important role in smallpox eradication. Also remember for your entrance exam that the World Health Organization declared smallpox eradication in which year? 1980. Apart from that, other scientist is the Robert Koch. The Robert Koch, it is known for the perfect bacteriological techniques. He introduced the staining method like the gram staining. So that allow for the visualization of bacteria under the microscope. Apart from that, he developed the method for obtaining pure culture technique. He used solid media like agar plates to isolate and grow the individual bacterial colonies. Apart from that, he set up criteria that is known as Koch postulate. So that are the series of principles they are used to establish a causal relation between microorganism and the particular disease. So that we will gonna discuss that just in a few minutes. He also focused on the culture and the lifestyle of the anthrax bacilli. So he also demonstrated that it was caused by bacteria. Anthrax was caused by Bacillus anthracis. So his contribution, it laid down the development of the anthrax vaccine. Apart from that, he also discovered the tuberculous bacteria that is responsible tuberculosis, that is the Mycobacterium tuberculosis. He also identified the Vibrio cholerae, that is the causative agent of the cholera. So it was Robert Koch who laid down the foundation for the modern microbiology and understanding of the infectious diseases. So the most important he gave was the Koch postulates. So let us discuss about the Koch postulate. Apart from that, he was given a Nobel Prize in the year 1905 for his research on tuberculosis. That is important that you have to remember. Now, what are the Koch postulates? So Koch postulates, they are just a set of principles that are used to determine if specific microorganism is the cause. Specific microorganism is cause of the particular infectious diseases. So the first postulate is the constant association. What does that mean? The microorganism, it must always be found in individual with a disease and should not be present in the healthy individual. Second is the isolation. The microorganism must be isolated from the diseased individual and grown in pure culture. Third is a reproduction of disease. So when this pure culture is introduced into healthy laboratory animal, it should reproduce the same disease. Fourth is re-isolation. So the same microorganism, it should be rediscovered from the laboratory animals after reproducing the disease. So these postulate helps to establish a clear link between microorganism and specific disease. Now let us again go through the cause postulate with an example. Example is the tuberculosis and the mycobacterium tuberculosis. The first was constant association. So in an individual with the active tuberculosis, the mycobacterium tuberculosis, it is always found in their lungs or in the sputum. It is not found in the healthy individual. So that is known as constant association. The second is isolation. Suppose a scientist, the microbiologist, they take a sample of lung tissue or sputum from a patient with active tuberculosis. From this sample, they will isolate the mycobacterium tuberculosis in a pure culture in the laboratory. So that means they can grow the bacteria by itself away from any other microorganism. So that is known as isolation. Third, to confirm that mycobacterium is the cause of tuberculosis, researchers introduced the pure culture of this bacteria into healthy laboratory animal, like the guinea pig. So as a result, the laboratory animal will develop tuberculosis symptom similar to that seen in the humans. So that is known as reproduction of the disease. And last is the re-isolate. So from the laboratory animal that developed the tuberculosis, the scientists can re-isolate the mycobacterium in a pure culture. So confirming that it's the same bacteria that is causing the disease. So that is re-isolate. So these are the coach postulates that you should know and remember. Apart from that, let us discuss other scientists. Paul Ehrlich, it is known as father of chemotherapy. So for entrance exam, remember who is known as father of chemotherapy? It's the Paul Ehrlich. So he introduced a drug for the treatment of syphilis. So other was Alexander Fleming, who discovered substance that is penicillin notatum. So who discovered penicillin notatum? It was Alexander Fleming. Who introduced antibiotic streptomycin? It was Waxman. So the Waxman, he discovered the antibiotic streptomycin. So let us now discuss the morphology and physiology of the bacteria. So we have a prokaryotic cells and a eukaryotic cells. So they are the two major categories of the cellular organisms. Prokaryotic they are simpler in structure, so they lack a true nuclei. So their genetic material, it is located in the nucleoid region that is not enclosed by a membrane. The prokaryotes, it lacks the membrane bound organelles like mitochondria, endoplasmic reticulum, Golgi apparatus, and a true nucleus. So they all is not seen in a prokaryotic cell. The prokaryotic cell size, they are smaller in size, ranging from 1 to 5 microns. So how do these prokaryotes reproduce? They reproduce asexually through the process that is known as binary fission. That is division of the one cell into two identical daughter cells. So prokaryote, they can either be autotrophic, that it means they can make their own food through photosynthesis, or they can be heterotrophic, that means they obtain nutrients from the other sources. Whereas the eukaryotes, they are more complex. They have a true nucleus enclosed by a nuclear membrane. So their genetic material, it is organized into multiple linear chromosomes. So they contain the membrane bound organelles, all the nucleus, endoplasmic reticulum, mitochondria, lysosomes, Golgi apparatus are seen. So these all structures are seen in eukaryotic cells. Regarding their size vary from 10 to 100 micrometer. Regarding their reproduction, they can reproduce both asexually and sexually. Sexually involves the fusion of gametes, that is through the sperm and the egg. So eukaryotes can be autotrophic as in plants and heterotrophic as in animals and fungi. So eukaryotes involves a diverse range of organisms like animals, plants, fungi, etc. So these are some of the key difference between the prokaryotes and eukaryotes that you have to just remember for your exam. Now let us discuss the type of microscope that we use to visualize the microorganisms. In microbiology, we can use is number one is the compound microscope. So it's the standard tool in microbiology labs for observing the stained and unstained microorganisms. Other type of microscope we can use is fluorescence microscope. So it is crucial in microbiology for studying the fluorescently labeled cells or the cellular structure. So it is used to visualize the specific microbial components or the traces or trace the presence of the particular microorganisms in the sample. Third is the phase contrast microscope. So this microscopy, it is ideal for observing the microorganisms without staining. So it will enhance the contrast and allow for the visualization of the cellular organelles. So it is effective for observing live and unstained specimens. Now, what is the principle of phase contrast microscopy? So remember that the phase contrast take advantage of there is a difference in refractive index of various material within the specimen. like the bacterial cells and the surrounding medium, usually the culture media or the microscopic slide. So when the light pass through these material, it can experience a phase shift or delay due to difference in the refractive index. So for entrance exam, you have to just remember that the principle of phase contrast microscope is due to the difference in the refractive index. So in phase contrast microscope, the phase difference between the light that pass through the object, that is the bacterial cells, and light that pass through the surrounding medium are converted into differences in the intensity of light. So this conversion it is achieved through the specialized optical component that includes the phase plates and the annular rings. So the phase difference in specimen they are transformed into the light and dark contrast in the final image. So areas of specimen that have different thickness are visualized more distinctly. So that enhances the visibility of the internal structures within the cells and make the bacterial cells clearly visible against their background. So remember, phase contrast microscopy, it is used for studying live it is used for studying the live microorganisms, as it allows for the real-time observation without the need for staining that can be harmful to the cells. So this technique provides a non-invasive and non-destructive method for visualizing the biological specimens. The fourth one is the dark field microscopy. So the dark field microscopy, it is used for visualizing the live unstained microorganisms. So that provides excellent contrast by illuminating the specimen against the dark background. So the dark field microscopy, it is an optical technique that uses to enhance the contrast in microscopy, making it particularly useful for visualizing the objects that are difficult to see under the standard bright field illumination. Now what is the principle of dark field microscopy? So under the bright field microscopy, where the specimen it is illuminated from below, the dark field microscopy it uses a specialized dark field condenser. So this condenser directs the light at oblique angle so that no direct light it enters the objective lens. So the result is that the light rays from the condenser only strike the specimen from the sides and scattered or reflected by the specimen. So that if there are structures in the specimen, they scatter light, creating contrast against dark backgrounds. So this scattering of light, it will produce that is structures that are dark field. So whereas the specimen they will appear self-luminous against the dark background. So dark field microscopy it is particularly used for observing slender or transparent objects that are not visible under bright field illumination. It is used in microbiology to observe microorganisms with spiral or helical shaped structures like spirochetes. That is the point that you have to remember for entrance exam, that dark field microscopy it is used for visualizing the spirochetes. So what it does, it improves the contrast and visualize the objects that are challenging to see under the standard bright field illumination. Other microscope we can use is electron microscope. Other is transmission electron microscope. It is used for visualizing the high resolution imaging of the internal structure of microorganisms. Other is scanning electron microscope that allows for visualization of the 3D structures with the depth of field. Other is eighth is the confocal laser scanning microscope. So it is used for the 3D images and analyzing of microorganisms, for understanding the cell structures and studying the biofilm. So these are the microscope that we can use in the field of microscopy. So now let us move to the stain. What are the stains that we use in the microscopy? We can use is the simple staining that includes methylene blue or the basic fusion. So simple stains involving a single dye like the methylene blue or the basic fusions to stain all the bacteria in the sample. So this impart the same color to all the bacterial cells, providing contrast for visualizing the cell morphology and the structure. So methylene blue and basic fusion, they are most commonly used stains in the microbiology. Methylene blue, it is a synthetic dark blue or greenish blue dye. It is used for methylene blue. Remember, it is used for staining the biological specimens, including bacterial cells. So methylene blue, it is used in simple staining to increase the visibility of the bacterial structures under the microscope. So when used to stain bacteria, methylene blue can provide contrast by coloring the bacterial cells while leaving the background colorless. So it is a basic or positive charge stain. So it tends to bind to the negative charge structures within the bacterial cells. Whereas basic fusion, it is bright red or magenta dye. So it is used in a gram staining to differentiate gram negative and positive bacteria. So it is used in counter staining the component of the gram stain. So in gram staining, the basic fusion, it is used as a counter stain after the crystal violet and iodine that will impart color to the gram negative bacteria. Other is the negative stain. Negative stain uses the dye. It uses the dye like Indian ink and nigrosin. So remember, the negative stain uses Indian ink or nigrosin. That is what you have to remember for the entrance exam point of view. So negative staining, the dyes like Indian ink or nigrosin, they are used to stain the background. Whereas the bacteria, they are colorless. So the unstained bacteria stand out in contrast against this background. So the negative staining, it is used for visualizing the bacterial capsule. So it is used for visualizing bacterial capsule. Again, for entrance exam, you have to remember this much of knowledge. And very slender bacteria like spirochetes that may not take the simple stains. So negative staining, it is a microbiological staining technique used to visualize and study microorganisms, particularly those who are very delicate to withstand the harsh treatment of the traditional staining method, like the heat, fixation, and chemical dyes. So the third is the impregnation method. So for entrance exam, you have to just remember that involves this method involves the deposition of silver on the surface of thin bacterial cells that is used to thicken them, that makes it visible under the microscope. So they are used for determining the microorganisms like spirochetes and bacterial flagella. The next stain is the differential stain. So differential staining, it imparts different colors to different bacteria. So the most commonly used stains that we use for in this are the gram stains and acid fast stain. So in differential staining, we use more than one stain. It imparts two or more different color to bacterial cells. Example are the gram stain and acid fast stain. So that is the difference between the simple staining and the differential staining. Now what is the gram staining? Very important for your entrance exam point of view. The gram staining impart different colors to different bacteria. So gram staining was developed by Christian Gram, who is used to classify bacteria into broad groups, gram positive and gram negative. The procedure involves primary staining with the dye, application of the iodine, decolorizing with an organic solvent, and counter staining with a dye of a contrast color. So the gram positive bacteria, they will retain the primary stain, that is the primary stain was crystal violet, whereas the gram negative bacteria are decolorized, take the counter stain. Counter stain was safranin, appearing red. So the gram positive bacteria retain the primary stain and appear violet, and the gram negative bacteria, they are decolorized and take the counter stain appearing red. So that's the principle of the gram staining. So gram staining technique, it relies on the difference in the structure and the composition of cell wall in the bacteria. The gram positive bacteria, the gram positive bacteria, they have a thick peptidoglycan in their cell wall that retain the primary crystal violet stain. So this thick layer of peptidoglycan, it forms a complex with the dye, making it difficult to wash out, whereas the gram negative bacteria, they have a thinner peptidoglycan layer. So the crystal violet, it is not retained as effective and can be washed out during the discolorization step. So the crystal violet is not retained. So after decolorization, the gram negative bacteria, it will take the counter stain, that is usually the safranin. Whereas the gram positive bacteria, they retain the violet stain. So we wash with the after. So let us discuss the procedure. First of all, we prepare a bacterial smear. We cover the smear with a primary stain, typically the crystal violet or gentian violet. So allow it to stand for about 1 minute So these steps will stain all the bacterial cells in sample with the violet dye. The next step is the iodine. Apply a solution of iodine to the smear. So the iodine will form a complex with the crystal violet, enhancing its retention by the bacterial cells. So the next step is decolorization. We use is 95% ethyl alcohol. So rinse the slide with organic solvents such as ethanol or acetone. So this step is important as it differentiates between the gram-positive and the negative bacteria. So the gram-positive bacteria will retain this crystal violet, while the gram-negative bacteria, they are decolorized and they will lose the violet stain. The next step is counter staining. So apply a contrast dye using usually saffron or fuchsia to smear. So the counter staining will impart a contrast color to the decolorized gram-negative bacteria. So that is the red color. So rinse the slide gently with water and blot it dry with bibulous paper. So the next stain is let us discuss the next stain is the acid fast stain. So that is used for to differentiate the acid fast bacteria, that is the Mycobacterium species, from the non-acid bacteria. So negative staining, it is so this method is used to identify the acid fast microorganisms like the Mycobacterium tuberculosis that causes the tuberculosis. Other is the Albert stain. Albert stain So Albert stain, it is used to highlight the metachromatic granules in the bacteria, especially the Corynebacterium diphtheriae. The metachromatic granules, remember, they take up bluish purple color when stained. So the Corynebacterium diphtheriae, it is characterized by slender bacilli with the metachromatic granules, also known as polar bodies, volutine, or it is known as Babe's Ernest granules. The key point that you have to remember for your entrance exam. Albert stain, it is used to demonstrate these granules, aiding in the identification of these bacteria. So Albert staining, it is a type of differential stain that is used for staining high molecular weight polymers of polyphosphate known as the metachromatic granules. These are high molecular weight polymers of polyphosphate that are found in Corynebacterium diphtheria. They are also found in Yersinia pestis and Mycobacterium species. So you can get a question, the metachromatic granules, they are present in all of these species except, or you can get one of the question, they are present in Corynebacterium diphtheria, or they could be seen in Mycobacterium species. So very important for your entrance exam, what are Babe's Ernest granules, metachromatic granules, all these are important. Now let us, coming to the bacterial shapes, let us discuss the various shapes of first of all, they are the cocci. They are the spherical or oval in shape. The coccus, it is derived from Greek word meaning berry. So they can occur singly or in clusters, like they can occur in pairs, where they are known as diplococci. Chains, they can occur in chains, then they are known as streptococci, or group of four, like tetrads, or clusters resembling grapes. like in case of Staphylococcus aureus. The Vibrio, they are characterized by their comma-shaped or curved-shaped appearance. So they are curved rod-shaped. So they are known for their vibratory motility. Other is Spirilla. It is a rigid spiral shaped bacteria. They have a helical or corkscrew like appearance. Actinomycetes, they are filamentous bacteria with the branching structures. So they radiate outerward like a ray of sun. So that is why they are known as a ray fungus. Ray fungus, they are named as actinomycetes. Another MCQ that you have to remember, another point that you have to remember for your entrance exam point of view. Mycoplasma, they are unique bacteria, they lack cell wall. So mycoplasma, remember for entrance exam, they will lack cell wall. So they can appear as round or oval bodies. So when cell wall synthesis is disrupted due to factors like antibiotic exposure, the bacteria may lose their shape and form. What that is known as protoplast. Spheroplast or L form. Now let us discuss the structure component of the gram-positive bacteria. The gram-positive bacteria, it is composed of thick layer of peptidoglycan. Which can be around 15 to 80 nanometer thick. So this layer, it would provide the support and rigidity to the bacterial cell. The peptidoglycan, it is a complex polymer that is made up of two sugar molecules, that is NAC and NAM, N-acetyl glucosamine and N-acetyl muramic acid. So these sugars molecules alternate to form long chains. So the peptidoglycan chains, they are cross-linked by peptide chains, creating a three-dimensional scaffolding structure. Apart from that, they contain important structure that is teichoic acid. Multiple questions have been asked. Teichoic acid is present in the gram-positive bacteria. And what is its role? Its role is in structural and it provides antigenic property to the cell wall. So teichoic acid, it can contain ribitol, all glycerol polymer. So these polymers, they are linked to peptidoglycan in the cell wall. So there are two types of teichoic acid in the gram-positive cell wall: the cell wall teichoic acid and membrane teichoic acid, that is known as lipoteichoic acid. So the lipoteichoic acid is linked to membrane glycolipids, and cell wall, it contributes to overall structure of the cell wall. So teichoic acid, generally you have to remember that the teichoic acid, it is a surface antigen in gram-positive bacteria and it is involved in recognition of bacterium by the host immune system. So the composition of cell wall in the gram-positive bacteria, it differs from the gram-negative bacteria. So first is the gram-positive, they have a thick peptidoglycan layer, and second is they have a significant amount of teichoic acid that contributes to overall structure and antigenic property. In gram-negative bacteria, you will see lipopolysaccharides. The outer membrane, lipoprotein, and the peptidoglycan layer. The lipopolysaccharides, it is the major component of the cell wall of the gram-negative bacteria. So lipopolysaccharides is responsible for, remember, endotoxic activity. So which one is responsible for endotoxic activity? It's the lipopolysaccharides. So lipopolysaccharides, it has, remember, it has three zones: region 1, region 2 and region 3. What is region 1? It consists of O polysaccharide. Region 2 consists of core polysaccharide, and region 3, it is glycolipid. Or lipid A. So which portion is responsible for endotoxic activity? It's the region 3, that is glycolipid, lipid A component. It is responsible for the endotoxic activity of the gram-negative bacteria. That involves these activities that will include the fever inducing. So they are fever inducing, causes tissue necrosis, stimulation of the B cells, and immunoadjuvant properties. The O polysaccharide determines the specificity of the O antigen. So that is important for identifying the different strain of the gram-negative bacteria. Region 2, that is the core polysaccharide. It serves as the structural component of the lipopolysaccharides. So for entrance exam, you have to remember that region 3 is responsible for endotoxic activity, and it is known as glycolipid portion or lipid A portion. So the endotoxic activities, they are the pyrogenicity, that means fever inducing, tissue necrosis, anti-complementary activities, stimulation of beta cells, immunoadjuvant activities, etc. The outer membrane of the gram-negative bacterial cell, it is known as outer membrane. It contains various proteins like the outer membrane protein that involves the formation of transmembrane pores that are known as these are the porins. So these porins will act as a diffusion channels for small molecules, and also function as receptors for certain receptors for bacteriophage. The peptidoglycan layer here, this layer is it is thin and it is bound by the lipoprotein. The lysozyme, it is an enzyme that can break down the mucopeptide link in the cell wall and can lyse the susceptible bacteria. Remember, the protoplast, they are formed when lysosome attacks on the gram-positive bacteria in a hypertonic solution. So protoplast, they are formed when lysosome it act on the gram-positive bacteria in a hypertonic solution, whereas the spheroplast, they are formed when the lysosomes act on gram-negative bacteria. So they are similar to protoplast, but some cell wall material is retained. So what is the main difference between the gram-positive bacteria and the gram-negative bacteria? The gram-positive bacteria, they have a single layer smooth cell wall. Gram-negative bacteria, they have a double layer cell wall. The thickness of cell wall is 20 to 80 nanometer. Teichoic acid is present. Peptidoglycan is thick layer, whereas it is thin layer in the gram-negative bacteria. Lipopolysaccharides, remember, it is not present in the gram-positive bacteria, present in the gram-negative bacteria. Outer membrane is not present in gram-positive bacteria. Lipid content is very low. Lipid content is around 20 to 30%. The gram-positive bacteria, they are very susceptible to antibiotics, and gram-negative bacteria are resistant to antibiotics. So this one is the key difference between the gram-positive bacteria and negative bacteria. Apart from that, there is the cytoplasmic membrane. Is the membrane that lines the inner surface of the cell wall and separates it from cytoplasm. So the cytoplasmic membrane will act as a semi-permeable membrane that will allow flow of metabolites, passage of molecules like nutrients, waste products between the cell interior and external environment. So this ability to pass through the membrane, it is determined by the size of molecules and presence of enzymes like permeases. The cytoplasmic membrane, it is composed of lipoproteins. The eukaryotic cells, unlike eukaryotic cells, bacterial cells lack sterols in their cytoplasmic membrane. The only exception is the mycoplasma. Remember the mycoplasma, it may contain sterols in the membrane. Another important MCQ point to remember. The cytoplasm, it contains various structures like ribosomes, mesosomes, vacuoles, etc. The ribosomes, they are center for the protein synthesis. The ribosomes, they are integrated into the linear strands of messenger RNA to form polysomes. So these arrangement it is that allow for the efficient protein synthesis. The mesosomes, remember you can get a MCQ how mesosomes are formed. They are formed by invagination of the plasma membrane. Mesosomes, they are more prominent in gram-positive bacteria compared to the gram-negative bacteria. So the mesosomes, they serve as principal site for respiratory enzymes, similar to role of mitochondria in the eukaryotic cells. Apart from that, you can get is intracytoplasmic inclusions. These are the granules that are found within the bacterial cytoplasm. The granules may be volutin granules that are the metachromatic granules, polysaccharide granules, lipid inclusions, volutin granules. They are also known as metachromatic or Babes honest granules. The polysaccharide granules, they can be they are the storage products in the bacterial cells. They can be visualized by staining with the iodine. While the lipid inclusions, they can be stained with the fat soluble dyes like the Sudan black. Nuclei, the bacterial nuclei are referred as nucleoids, and they differ from the nuclei of the eukaryotic cells. The bacterial nuclei, it can be demonstrated by special technique like the ribonuclease hydrolysis for staining for the nuclear material. They can be visualized under the electron microscope. The bacterial nuclei, they don't have nuclear membrane. So bacterial nuclei, they will not have a nuclear membrane or the nucleolus that is present in the eukaryotic cells. The bacteria genome, it consists of single-stranded circular molecule of double-stranded DNA. So bacterial genome, it consists of single circular double-stranded DNA that is not associated with the basic protein. Unlike the eukaryotic cells, the bacterial chromosomes, they are haploid. That means that they have one copy of genetic material. They replicate by process that is known as simple fission.